Welcome to BioVivacious. BioVivacious is a YouTube channel dedicated to clear fundamental concepts in biosciences. Today we are going to take up a topic, mucopolysaccharides. They are also known as glycosaminoglycans, and all these molecules have about 5% proteins and about 95% carbohydrates. They are also known as proteoglycans, and these are substances that are present in living system, especially in human being, in animal system, vertebrates, invertebrates, and as well as in bacteria. They have not been so far reported in plant species. When we look at mucopolysaccharides, it also has got a physiological significance. We will be looking at that at the end of it. Mucopolysaccharide is a heteropolysaccharide. We understand what is the meaning of heteropolysaccharides. It means different monomeric units are being used in order to make a mucopolysaccharide. Generally, if the two units, sugar units that are used in a mucopolysaccharide, one is a uronic acid, the other one is N-acetyl glucosamine. So you have N-acetyl glucosamine and a uronic acid. So from the name we understand it has got a highly negative charge present in all mucopolysaccharides. So these units are repeated with a mild variation. Some of them will also be sulfated. That gives a very high negative charge to mucopolysaccharide. These mucopolysaccharides are largely abundant in ligaments, in synovial fluid, in vitreous humor, in the tendons, in extracellular matrix. These mucopolysaccharides are largely found. Being in extracellular matrix, the mucopolysaccharides are responsible for giving a particular shape to a cell. We are going to look at each of the mucopolysaccharides. All mucopolysaccharides have a common structure except hyaluronic acid. And this structure is, these are embedded in the cell plasma membrane. Assume this is the plasma membrane. So in the mucopolysaccharide will have a central core, the 5% protein that we are discussing about. So this will be the central core of the protein. So this is a protein. And as we know, the protein cannot be a branched structure. So, but carbohydrates can get attached to protein, especially wherever there are serine residues, the carbohydrates can get attached. To the hydroxyl group of the serine, a carbohydrate, here it may be a mucopolysaccharide like dermatan sulfate, or this may be a kind of a chondroitin sulfate. So this is how mucopolysaccharides are arranged in the extracellular matrix or embedded in the plasma membrane of a cell. This is going to be the general structure of a mucopolysaccharide. Now we will look at the structure of each of these mucopolysaccharides in detail, and we will also look at what are the functions these mucopolysaccharides carry out. This is a summary table that explains the kind of monomeric units that are found in mucopolysaccharides. So therefore you look at the glycosaminoglycans, there are mainly 5 of them that we are explaining in this video. For example, hyaluronic acid, if the 2 monomeric units are glucuronic acid and N-acetyl glucosamine, that will be linked by a beta-1,4 linkage. When you look at heparin sulfate, it is glucuronic acid linked by alpha-1,4 linkage to N-acetyl glucosamine. Chondroitin sulfate will have glucuronic acid beta 1,3 linked with the N-acetyl galactosamine. That is to be noted. Dermatin sulfate has got another form of glucuronic acid, that is iduronic acid, with the beta 1,3 linkage with the N-acetyl galactosamine, and finally keratin sulfate will have galactose linking with the N-acetyl glucosamine with the beta 1,4 linkage. In the first glycosaminoglycan we are going to look at is hyaluronic acid. As we have seen in the previous table, hyaluronic acid is made up of glucuronic acid with the N-acetyl glucosamine. So if the glucuronic acid is linked with the carbon number 1 with the carbon number 3 of N-acetyl glucosamine. So this is beta 1,3 linkage. Hyaluronic acid is the only glycosaminoglycan identified so far which is not connected to a protein. These are free. These are found in the vitreous humor. It is found in the synovial fluid. It is found in the joints. And the main function of hyaluronic acid, it will act as a lubricant. Now, as we know, all the glycosaminoglycans have a capacity to absorb water. So a kind of a water shell form around this molecule, and that will make them to act like a lubricant. So therefore it will help in the lot of free movements in the joints. We know that when people get old, when they are unable to walk properly, when there is a friction that is being caused in between the cartilages, the kind of medicine that are proposed is having glucosamine. Now, this is a molecule, this is a disaccharide unit. This disaccharide unit will be repeated maybe about 5,000 times. This disaccharide unit, which is joined by beta 1,4 linkage. Disaccharide unit here, another beta 1,4 linkage will be there. So these disaccharide units are repeated almost 5,000 times. So therefore the molecular weight of this molecule will be very high. It is estimated that the molecular weight will range from 10 raised to the power 6 to 10 raised to the power 7 Dalton. So we have an enzyme in our body, it is known as hyaluronidase. Why do we need this enzyme? Hyaluronidase will help in cleaving this bond and making, it makes it a very thin layer. So as a result of which biomolecules are able to pass through. For example, as we get older, if the amount of hyaluronidase will decrease, that will make the hyaluronic acid very thick, as a result of which nutrients may not be able to be supplied to the synovial fluid or to vitreous humor. This can also lead to impairment in vision. In the next glycosaminoglycan we are going to look at is heparin. Heparin, as we know, it is an anticoagulant which is found in blood, in liver, in lungs, in mast cells, everywhere heparin is found. This is a molecule with about 17 kilodalton as the molecular weight. This molecule is made up of these two monomeric units. One is L-iduronic acid. So remember, we have put the COOH inside. This is iduronic acid. And the next molecule is N-sulfoglucosamine 6-phosphate. In heparin and heparin sulfate, you will find plenty of sulfation happening, which was not observed in the case of hyaluronic acid. Hyaluronic acid is not at all sulfated, whereas now onwards all the glycosaminoglycans that we will come across will be sulfated. Also observe that the kind of linkage which will connect iduronic acid with the N-sulfoglucosamine 6-sulfate will be alpha 1,4 linkage. So therefore this is also a very unique glycosaminoglycan with alpha 1,4 linkage. This is the only glycosaminoglycan with an alpha 1,4 linkage that has been identified so far. As I said, this will act as an anticoagulant that will bind with the antithrombin number 3, and it will prevent coagulation of blood. The next glycosaminoglycan we are going to look at is chondroitin 4 sulphate. So in the case of chondroitin 4 sulphate, the monomeric units are, this is glucuronic acid, and this is N-acetyl glucosamine. Now the glucosamine is sulfated at the 4th position, so therefore it is chondroitin 4 sulphate. And the kind of linkage is already shown in blue color. It is between carbon number 1 and carbon number 3, that is a beta 1,3 linkage. The molecular weight of this substance will vary somewhere from 5 to 50 kilodalton will be the molecular weight of this substance. So this will keep repeating, and that forms a linear chain, and that will be linked to a protein through either serine residue. So this is one of the most abundant glycosaminoglycan that is found in living system. So they are found in the heart, they are found in the aorta, they are found in the synovial fluid. Everywhere this chondroitin sulphate is found. Now this chondroitin sulphate, there are varieties of chondroitin sulphate depending on where the sulfation occurs. This is in the 4th position if the sulfation has occurred. If it is occurring at the 6th position, a sulphate is added here. So that becomes chondroitin 6 sulphate. So similarly, there are chondroitin sulphate A, B, C, D, E, depending on where sulfation occurs, varieties of chondroitin sulfates have been identified. Another glycosaminoglycan which we are going to discuss today is dermatin sulphate. Dermatin sulphate is found in skin, in heart, in the valves. Everywhere this dermatin sulphate is found. The molecular weight of dermatin sulphate ranges from about 5 to 40 kilodalton. The basic building units of dermatin sulphate is hyaluronic acid and N-acetyl glucosamine. They are linked through beta 1,3 linkages. So dermatin sulphate also gives a kind of a protection especially in the skin. The final glycosaminoglycan which we are going to discuss as part of this video is keratin sulphate. Keratin sulphate, as you know, in the basic units of keratin sulphate, this is a galactose. This is a galactose in the beta conformation, and that is linked to N-acetyl glucosamine, which is sulfated at the 6th position. The kind of linkage is beta 1,4 linkage. This keratin sulphate is present in all the weight-bearing tissues. For example, like ligament, tendons, they are present in cornea. So these are the places keratin sulphate is present. This also has the capacity to absorb water, and it hydrates in the skin. So after looking at all the glycosaminoglycans, let us look at what are some of the common functions of these glycosaminoglycans. Number one, they will give structural integrity to our system, our cells. They help in motility of cells. Some of the glycosaminoglycans are involved in cell adhesion, they are involved in chemotactic factors, they are involved in the growth factors, they are involved in cell adhesion. All these are some of the functions the glycosaminoglycans play. They have been implicated in the apoptosis in the recent literature. Accumulation of mucopolysaccharides, that is, if there are enzymes that are lacking in the degradation of these bonds, the mucopolysaccharides will get accumulated in our body. If it is getting accumulated in our body, it leads to a disorder known as mucopolysaccharidosis. So this is because enzymes are deficient in breaking down these molecules.